lecture series, and it's a great pleasure to introduce Philipp Löffler, postdoc student in my group. And he has a long history, and it's actually this year that he has his 10 years anniversary of receiving the bachelor degree in my group. And then he left me for quite many years to pursue his master studies and his PhD in the Flint Center, and he was working on protocells, and he was working on lipid nanoparticles, on RNA replication in a sort of in a primordial fashion. And he's a good chemist, but he's more than that, since he is one of our nanobioscience students. So he likes physics, he likes biology, he likes chemistry, and he has a completely different mindset to approach problems than a pure chemist would do or pure biologist would do. And I hope he will show us some of these aspects in his lecture, which is about bio-inspired chemistry, can we engineer self-assembly lines at the nanoscale. And please, Philipp, the scene is yours.
Thank you, Stefan, and thank you for inviting me to give this talk. I will try and walk you through as good as I can to give some principles, what the self-assembly is a little bit, what the nanoscale is a little bit, and what we are trying to achieve by mimicking it. Well, first of all, it's great to see all of you here, such a filled room, and I hope we can have a good time with these slides. Alright, so I don't know if many of you thought about this, what is this assembly lines, and then there's a self here. So assembly lines we all know from the good old Henry Ford, and production nowadays. But a self-assembly line would be one that can act without the interference of chemist or worker, so on. So that's the theme I try to design the talk from. First of all, none at this university or any other university has invented nanotechnology that has nature already many billion years ago, and if kind of try to walk you through the scales a little bit. So if we are in the nanometer scale here, then we have a tennis ball at 10 to the 8 nanometers, which is many nanometers. Just below the limit where we can see with our bare eyes the range of micrometers or tens of micrometers. It's the range of where biological cells are. Below that we find the building blocks of life. Nucleic acids, proteins, small sugar molecules, lipid molecules, which are here forming a nanoparticle. We study many different nanoparticles, from the smallest that are micelles we use to, that's formed from soap molecules and which we use to wash clothing with. Liposomes that constitute membranes for all biological cells, basically, or the scaffold for it. Then there are synthetic ones, dendrimers, gold nanoparticles, other metal quantum dots, and what you could not think of, polymer particles, and so on. So, with nanotechnology, we mean something that operates in the range between 1 to 100, maybe 1,000 nanometers. So, in the size and length scales of life, basically. The same scales where biology operates, where all of biology's machines operate. Because if you assemble things at the nanoscale, then you can basically do many, many processes in a very small environment, which is the cell. So, and in nature and in chemistry, one very fundamental principle for organizing all of these things at the nanoscale is self-assembly. Because when we get, well, human technology has actually developed the skills to manipulate individual atoms or proteins or cells, but we can only manipulate one at a time. If we use self-assembly, then the properties of molecules force them to behave in a way that gives them a certain structure over time. Here I show actually a dissipative particle dynamic simulation of some lipids that self-assemble in silico to form spherical shells, which I will refer to as liposomes or vesicles later on. And this is a process that in this simulation took 2 real milliseconds, probably many hours of computer time. But this is something that occurs every single time when chemicals with the right properties are mixed together. And this is what life uses ubiquitously all around its processes. And one other, among many examples I wanted to mention is, of course, nucleic acids. Here is also kind of a coarse-grained model just to illustrate how nucleic acids can dynamically form famous double helix, maybe on this blue strand here, making base pairs and twisting the structure, and another strand that also can bind here to the blue one, can interact with the green one, displace it maybe. So this is also a different kind of self-assembly. But what's important of this one is that this is dependent on a more higher level of organization. In this case, the sequence of the DNA, whereas this is dependent on a monomer level, on the level of a single molecule having properties for self-assembly with molecules that are alike. So we have kind of molecule-based self-assembly, and then we have the more advanced form that nature really loves is the sequence-based self-assembly, or self-assembly based on a certain property of a biopolymer. So, going back to assembly lines, just an illustration I found from a patent database is for making a method for dehydrating potato flakes, which you then can sell in the freezer and so on. So this is a very simple scheme where you have many processes, and in the end you package it, and then it's basically done. And this is kind of how assembly lines work usually in our man-made factories. But nature has actually quite some advantage in terms of complexity, mainly because it processes everything in parallel in the same factory. So, and many of you might know the central dogma of biology, which is basically that every cell has DNA in its nucleus, which has the instructions for making an RNA, which carry the instructions for making a protein, and the protein then has some structure and some function, could be called a nanomachine actually. And there are of course also proteins that do all of these processes. So they replicate the DNA, they transcribe it into RNA, and RNA and proteins translate the information in the RNA into a protein. So, and for humans, yes?
Yeah, can I ask a question here? So if you go one slide back, we have this assembly line, and the whole purpose of the assembly line is to have some inputs, then there's many boxes, and then we have some outputs. And the critical feature of the assembly line is quality control, to make sure whatever I intend to create at the end has the quality standards I wish to have. And if inside there's a CNC machine which is fully automated, right, a robot doing the stuff which may be set for self-assembly as well, at the end I need quality control. I have some specifications at the beginning that I need to check if they are fulfilled at the end. How does that map onto your self-assembly line?
Alright, so for DNA replication, for example, there are enzymes that check if the copy is correct, and they are as good as checking a few letters and a few billion characters, basically copying a whole encyclopedia without basically one error. And this is of course advanced machinery inside the cells. For the proteins, it's much more difficult actually, because there is no way of proofreading a protein nanomachine that is already done. So there the question is, does it work or doesn't it work? I think the humanity has many problems with proteins that do not work exactly as they're supposed to, which then often is traced back to an error in the DNA or something called genomically inherited diseases. Yes?
Well, evolution would tell us that the biological assembly line has low quality management because errors occur, and that by the end of the day is then resulting in an evolutionary process, right? Is that adequate?
I would call it several orders of magnitude higher quality than what we can do in the lab usually. So at least at this scale, let's say for microscopic processes, man has evolved quite a lot of quality assurance, but at the scale of a nano, nature is very very strong. Yeah.
I mean, without being a biologist or molecular biologist, I would comment on that the complexity of proofreading scales with the complexity of the system, so the organism. Of course, bacteria needs a lot less proofreading than a human to function. So I guess the proofreading is scaling with the complexity of the system. And,
Yes, I want to mention one example that reminisces a little bit what we do or what we intend to mimic using phospholipid membranes. So here's a schematic of a cell that produces some proteins based on its DNA, which is stored in the cell nucleus, transported over the endoplasmatic, synthesized in the endoplasmatic reticulum, and then transported further on, and eventually released in a process called biomembrane fusion. And in this case, what is released here is also called exosomes, and the cell processes these to release a certain protein, in this case a trypsin that can be used to degrade our food in the intestine. So I would call nature or cells as a way or as able to do massive parallel production of nanomachines. And we are not there at all, but it would be cool at least to do parallel production of one kind of nanomachine in our laboratory. So this biomembrane fusion, this fusion of intercellular vesicles, or in this case they're called exosomes, as a chemist, I think of it as mixing the contents of two tiny reaction flasks, or pouring a reaction flask into a bigger system. In biology, this, apart from the example I showed, this occurs all the time in the synapses of the brain, where neurotransmitters are released through the same process. The machinery that operates this is called the snare protein complex, and it's a rather complex machinery. I'm not at all an expert on this, but just walking you through a little bit, it depends on protein domains that form, well, some of them are on the acceptor membrane, and one of them is on a vesicle that's supposed to fuse with the membrane here illustrated on a surface. And it's based on the formation of something called coiled coils between large peptides or large domains of this snare protein complex. And yeah, in the end, the two membranes are brought together in such close proximity that they will rupture. And nature has evolved also some parts that go inside the biomembranes and really can open it. So it's a very well regulated and effective mechanism, which has been studied to great detail, but which is for a technological application, for at least our abilities, far too complex. So, to speak a bit more about liposomes as reaction flasks. So how tiny is the reaction flask? And what is needed to use it as a reaction flask. So first of all, we could fuse 1, 2, 3 different reagents or molecules into the same container. But we need to do it efficiently, because else it's like a sloppy chemist that pours half of it beside when you kind of want to do your reactions. And can we use something very reliable to program these steps beforehand? And DNA sequences give that opportunity. To give you an idea of the volumes that are involved, that if we have a liposome that's 100 nanometers, it's 400 septoliters in volume, or 0.4 attoliters, which is a very small volume. And if we can control it, chemistry at volumes like this, we can do chemistry on maybe thousands or millions or billions copies of a molecule, instead of very high numbers, moles, in our reaction flask. So you could browse many more reactions without using even a fraction of the material we would elsewise. Of course, this would be chemistry under crowded conditions, which means that if something is confined to a very small space and two molecules need to kind of bump into each other to react, this is actually an advantage. And non-bulk conditions, that means here that of course it would be chemistry in water, because liposomes, they form in water. But if water is confined to a small space, again it behaves differently. So certain reactions could occur that did not occur like in a normal reaction flask. Alright, so DNA programming. I don't know if I'm going through too fast, but the hypothesis is two liposomes, when they carry a complementary DNA, they can find each other spontaneously in the test tube. So that is our implementation of self-assembly, apart from that the liposome self-assemble. They self-assemble maybe around a certain molecule, and then we add some DNA that gives two liposomes the ability to find each other, or at least when they bump into each other they will stick. It's more like that. It's just in the length scales of liposomes, there can be many collision events in a few seconds. So they will find each other in the test tube. Right. So, but now if we want to design a molecular assembly line, as a chemist I like to assemble molecules, and usually we do that in a multi-step synthesis fashion, where I combine two reagents to form a third one, and then add a fourth to form the next product, and add a third to form the next one, and a fourth to form my final product, for example, for a three-stage synthesis. Here you see a schematic of liposomes that encapsulate, for example, A and B, the two reagents that we would like to mix together so they can react. On each liposome we have a DNA molecule, which then can find a complementary one on another liposome. So with the action of many liposomes, with the action of many DNA molecules, these will eventually fuse, and we can also introduce a recognition unit for the next population, and so on. So, for example, we could mix 4 things in a bigger reaction flask, using the sequence of DNA to encode it. I will go a little bit more into details how this works in a second, but this is just a general scheme that we use DNA to make 2 containers find each other, to fuse, which then triggers the reaction that can occur between A and B to form C, and so on. There are other ways to do that. You could, for example, instead of putting things on inside a liposome, you could directly couple them to DNA. And this has also been done in the literature. Recently, a very interesting system is that does basically peptide synthesis and fusing together a few peptide units, and at the same time recording all the events that happened with the DNA. Here, we could also record the events that happened, because that's basically the tags that are on the final compartment. And if we want to try a different chemistry, we do not need to make a new conjugate between DNA and between the DNA and our molecule that we want to react. We can simply encapsulate it in a liposome instead. So therefore we see liposome fusion as kind of a very flexible platform to move towards molecular assembly lines. And obviously nature has chosen the same, apart from the fact that it doesn't use DNA to control which compartments should find each other or which membranes should fuse. It uses many different proteins. So the snare protein that I showed you is just responsible for doing the fusion, and then there are other proteins and cofactors and recognition units that are responsible for the docking. But in the laboratory it has been shown that DNA can do both things. Alright. So let's go back to a little bit, zoom in a little bit more to the details of the system. I showed you these liposomes in a simulation, and if we take the schematic representation here and zoom in a little bit, then we need to take some DNAs that can find each other and link them to something we call a membrane anchor, which embeds into the biomembrane. It would, if you add it to a suspension of such liposomes, it would spontaneously find it and embed itself, and this way encode this vesicle population. So then if they are mixed, they can hybridize and force the membranes together. So in terms of a chemical structure, this anchor moiety looks like this. I shortened it a bit, but it's two C16 or palmitoyl chains that are linked into the phosphodiester backbone of DNA via a very small building block here, an amino diol, and a short linker. And in practice, for you that know DNA sequences, there are 17 nucleotides that form this zippering sequence, so to say, and they of course are made in our lab. So we do the organic synthesis of a building block. We study and characterize the formation of liposomes and the interactions of our DNAs with the membranes. For that we need some biophysics tools, binding studies, for example using surface plasmon resonance, but I won't go into details with that. And of course we need to synthesize the DNA and characterize it, which is now a fairly standard method, but is often in one lab. It's different from the flask-wise organic synthesis, as it actually is occurring also on a much smaller scale. So in order to study this liposome assembly and fusion in the lab, we need to combine different disciplines of science. When we look at two liposomes being cross-linked by DNA, in our case with the 17 nucleotide sequence that can form the duplex, it can only start to hybridize once the liposomes are maybe 10 nanometers apart. So this is already a fairly close spacing. But if the duplexes form, then the liposomes will end up very closely spaced, maybe around 2 nanometers, which is a space that is, a distance that is so short that the individual membranes can start mixing, and the individual molecules can kind of start exchanging between the liposomes, and eventually that causes fusion. Just statistically, it increases the likelihood tremendously. And here they are shown 3 DNA strands, and in reality it's probably at least 40 strands per liposome. In literature some have gone down to I think 13 or so. if you compare it to the snare protein complex, if it is in its natural environment having all the other factors present, then maybe 1 or 2 molecules is enough to fuse it. Here you need a little more, but it's actually still a few molecules compared to maybe 100,000 lipids in each liposome that it's kind of forcing together. Yes?
Those pictures, are they taken under the microscope?
No, I'm sorry. This is simply an illustration.
Okay.
We can. I will also go into this a little bit later, but we cannot see individual DNA molecules with a light microscope. With an atomic force microscope, you can do that, but that's not my specialty, so I will try not to go in there. But this is just a simulation of how the computer can find a configuration for these things, how closely they are spaced. So there might, in reality, be a difference. So we cannot measure these angstrom distances in these wet systems. For that we need kind of electron microscopes or atomic force microscopes, which typically look at static systems. So we cannot really follow this process in the test tube. We can only do that in silico. And this is, yeah. So this is meant as an illustration. But I will show some microscopy later. Oh, now, yes. So one thing I wanted to show you to illustrate that that many strands must be at work when we, for example, assemble giant liposomes here, is that they actually form kind of a large cross section. And dependent on the design and the length of the DNA and how closely it can put them together, they will fuse or not. But in the cross sections between such liposomes, there must be, at least for the giant liposomes, there must be thousands of DNA molecules. It's a different case when we go into smaller liposomes. All right. So, but using this zipper design, so where we try to make the DNA force the liposomes to go as close together as possible, then we have found results actually in opening of fusion pores, and in this case, if it could go there, it didn't go any further than this in this experiment, but in this case it would then form a much larger liposome. Alright, so how can we observe this? We have done experiments on small particles that we cannot see with a light microscope for a long time, but in the Bionec Center we were so lucky to collaborate with Aarhus University, and there they showed that if we encapsulate a dye of different color and 2 populations and then mix them together for some time and then look at the confocal microscope, then we can see some liposomes, or actually a significant fraction of the liposomes that contain both dyes, or that show up in both channels. This one up here is a bit weak in the green channel, I see, but yeah. So now we've already touched upon microscopy and the limits of it. Why not do that for small vesicles? It would probably look something like that on a microscope, because we cannot discern the individual particles in a light microscope, that is. In electron microscope techniques, the disadvantage again is that we need to kind of freeze or stain or do something else to a sample, so we cannot really see the live process. I mean, even for confocal microscopy, there's some time that needs to pass before you can actually visualize these particles, but that's technical details. Alright, so we want to fuse nanoscale compartments. So how can we tell? So I will try to introduce a few of the methods that we have used, mainly 2, but we can tell that the lipids of the individual liposome populations mix by using the Fred effect, so a transfer of excitation energy from 2 dyes that are in the membrane, which changes when 2 liposomes fuse. But I won't talk about that. A much more straightforward way for me is the mixing of contents, where we also use, again, a fluorescent dye that's entrapped in one of the liposome populations, but in such a high concentration that it's actually kind of really black and does not emit any light. But when it's diluted, it can start emitting light. We call this phenomenon self-quenching. It's just, if the dye concentration is high enough, no light can actually pass through it, probably either to excite or to emit any light. But when it's diluted, we can detect this as a fluorescent signal. Also, there are various techniques to monitor the size of liposomes. But as you might already guess, you cannot really see whether or not it's actually fused just by looking at the size, if we cannot really visualize the molecular shape of the things. Alright. One of these techniques that I really like, because it gives an absolute measure on how many particles are in a system, and gives an illustration of it gives a picture of how the individual particles have evolved over time if we do analysis at several times. Nanoparticle tracking is basically a microscope setup where you have the particles in a flow cell, and a laser shines through at a shallow angle. And you don't look at the individual particles, but you look at the kind of the scattering image that is created by the laser beam bouncing off the individual particles. So you can visualize nanoparticles, but you can only see them if they do not overlap. So you need to look at very dilute samples. So this could be a sample frame from a video that you take. So you make many frames of this for maybe 30 seconds or 60 seconds, and then based on the Brownian motion of the particles, one can calculate the size, not for the whole population, but for each and every particle that could be tracked for more than 10 frames in a row, for example. So this is, for us, a good method for keeping track of the particle size over time also. Many might know dynamic light scattering, which one also can use to obtain a size distribution over the particles that you made. But it's typically a more rapid method, but in our case this gives a more detailed picture of what's going on. Alright, so let's follow a fusion process with the nanoparticle tracking. So just for starters, I wanted to say that if you fuse two liposomes, so if you make 100 nanometer liposomes, that's of course an average, then you have encapsulated 0.4 attoliters of volume. If you fuse two liposomes, then the volume doubles. If you add a third liposome of the same size, the volume only is increased by a factor of 1.5, and the factor decreases as you go along. So that's, and therefore also at the same time also the radius does not double, or the diameter is not double for each fusion event to its spherical shape. Anyway, when we follow at several instances after mixing two populations with complementary DNA, if we follow the size distribution of our liposomes, then we can see that it first increases and then decreases, but settles at a higher level than before. And this, to us, reflects that the particles assemble into maybe higher assemblies before then they fuse together. And in this case, actually, the starting and end mean diameter corresponds to maybe that 80% or so of the liposomes actually did fuse. But we don't know for sure. So, going back to small vesicles. So now we're looking at the content mixing assay. And this is done using a fluorimeter. So instead of looking at the individual liposomes in a microscope, we put them all into kind of a glass chamber, cuvette, and we'll shine light on it, and then fluorescent light is emitted at a 90-degree angle. At least that's what we observe with a detector. And so if fusion occurs, we will see an increase in this emitted fluorescent light, as we call it. Yeah, and just I wanted to mention that only if the contents actually mix, then we see an increase in the fluorescence. If they just stay as they are or are assembled, then nothing happens. But unfortunately, the fluorescence can not only increase by dilution inside, but it could also leak to the outside of the liposomes. And so therefore, when designing these experiments, we need to make a control for it. And also an interesting control is if the DNA sequences of our zipper are not complementary. So here you can see some examples of how the fluorescence intensity evolves over time, and for our complementary systems we see a steep increase in the beginning until it kind of level off after some minutes. But all the controls remain fairly low. Actually, if you don't add any of our DNA to the surface, the leakage is higher than in presence of the complementary system, which causes them to fuse. So that means that the force that is exerted on the membranes doesn't force them to rupture or makes them more permeable to whatever we have in there. So something that's important when we want to do chemistry again. It's not so nice to have a reaction flask that has kind of a fracture and starts dripping over time. So, and with our system we could show that a system that actually spaces a little bit the DNA sequence from the surface, then we could observe up to 3 actually fusion events on one of the on the population that we monitor. So so we can actually quantitatively add volumes of new stuff to one liposome population. Alright, so going back to the assembly line. So I showed you this graph before, and now we have to try it in the laboratory. So can we actually do this several stages on? And usually we want a proof of concept before, so I didn't want to study also the reaction that's occurring inside, so I just follow it with the assay that I explained previously, just as the dilution will increase for every time we add a new population of liposomes. So, and the idea is that such a assembly line should work in a successive manner. That means when the first 2 liposomes fuse, then the one that brings the first reagent brings a recognition unit which allows the product population to fuse with the next reagent population. And in turn, the next reagent population carries the DNA strand that allows it to recognize the third reagent population. And in turn, we should mix these 4 reagents in a successive manner, each one dependent on the success of the previous one. Unfortunately, in our assay, for each step, as I told you, the dilution factor becomes less. So we have a factor 2 for the first, factor 1.5, and a factor 1.33, so for the third. So in this case, the assay is maybe not the best, but we can surely show that for each addition of a new reagent population, we see a new steep increase in the signal, or maybe for the third one not so steep, but it depends. It also is because every time we add a new reagent population, then only the ones that have fused in the first round can fuse on for the second round. So this might sound a bit complex to you, but it's, at least for me as a chemist, nothing as complex as if I were to study for example, the exosome cascade in biology. Yes?
Why is it that they can only fuse with the fused one next time, because there's still a green one?
Yeah, so this one is the population that we monitor. we can see because it has the dye. And then we fuse this population 1 here, and that one has 2 DNA on there. One that is able to recognize the first monitored population and is responsible for fusing, and then another one that does not pair to this one. So that the first 2 DNAs, they end up as a duplex on the same surface because the membranes, they go together. And so also this next one is transferred to this product population. And now it contains both kind of the spent DNA and a new one that can recognize the next one, container.
Yes, but do you wash the rest of the non-reactive away then?
No,
Can't it react with the free one?
But you can monitor it.
Oh,
That's
we can
true.
monitor it.
That's
Okay.
true. That's a very good good point. If we have some of those that haven't reacted with the first population, they can in principle react with the or fuse with population 2 in the background. That's very correct. And so therefore we are also interested that this process is very kind of effective, because else it prevents for the next step. Are there more questions to this? Right. So also in this case, we need to make a control where we simply fill everything with the self-quenching dye to see the signal increase that's only resulting from the dye leaking out into the outer medium. That also would decreases the concentration inside and gives a fluorescent concentration on the outside of the liposomes. Yeah. So, how good is our sloppy chemist? That we can tell from looking at each individual stage, kind of looking at how many of the possible liposomes have fused. So it's not maybe the perfect analogy from like pouring 2 flasks together. It's more like you pour billions of flasks together and then maybe half of them spill. Or at least here it's successful for 60%, and for the next stage, for the ones that made it to the second stage, it's around 80%, and then it becomes a little lower for the third stage, for various reasons that we could discuss. But in the interest of time, I will move on to my idea slide, and I think I have some time left. But with what I just showed you, with the technology that we now have, at least the proof of principle, it can become much better. We are puzzled to find some good applications for this in a kind of technological way. So I think it's one example of actually demonstrating that we can spatially control and also over time control when things happen at a nanoscale. And that could have several advantages for other branches of science. For example, many enzymatic reactions in biology occur inside the liposome or in the membrane of the liposome. So if we can pre-assemble reagents and enzymes in each population, then we could trigger the reaction just when we're ready to observe the reaction happening. This is kind of maybe a technical thing, but if you want to mix things in the liposome and then look at it in a microscope or in a fluorimeter, that passes time. So the reaction might have actually already occurred, or the conditions that we want to study are not the same anymore as in the beginning. So also what would be very important to do now as the next step is to actually do chemical reactions. Just simple catalyzed or non-catalyzed chemical reactions in a physiological conditions, but inside these entrapped volumes, programmed by our kind of DNA assembly line. There's a big hype about artificial cellular systems, where you actually try to recreate systems that have some of the properties as living cells do, the ability to self-replicate, the ability to take up nutrients and transform them to something useful. for their own machinery. There, of course, being able to fuse compartments together could be a good tool. And finally, I want to mention an idea that an old colleague of mine, Harold Fellerman, kind of came up with when we were at the Flint Center. He called it the container calculus, where you could actually make different operations with the encapsulated molecules, and in this way synthesize a target molecule or solve a mathematical or chemical problem. For example, does some sample contain molecule X, or does X and Y react? So could answer on chemical problems in kind of a real test tube implementation. Also, drug delivery. Several mimics of the snare proteins have actually been used to fuse liposomes directly to the skin of a zebrafish. So you could imagine that if you kind of pre-treat the tissue with a kind of recognition unit, DNA, you could add then some liposomal formulation that would kind of only go into the membrane of that tissue that you have pre-targeted somehow with the DNA. Alright, I'll go back to the slide afterwards, but let me just express my biggest thanks for all the possibilities that we've had here at the Biomolecular Nanoscale Engineering Center, where we actually work with all the different classes of biomolecules, like proteins, nucleic acids, carbohydrates, and lipids, make different combinations of them, which then show to have new properties. Like, for example, our DNA and lipid conjugates have the ability to fuse liposomes, something that actually does not exist in nature. DNA molecules that fuse liposomes. Or maybe we have just not discovered them, but at least there we have the possibility to create something new. And in this conjunction, I want to thank all the people that helped in this project for the liposome fusion, especially a group of Jørgen Kjems, Rasmus and Anders, who helped with the confocal microscopy, all the staff and students and postdocs, and also the laborants at SDU and the group of Jesper Wing. Been great help in DNA synthesis and doing important discussions on how to make the correct controls, for example, and if everything is occurring as it should. And of course the focus group, especially Oliver, who kind of was the starter of this fusion project here, and Alex, who does a similar thing with the peptide nucleic acids, and Louise, who makes all our liposomes, makes all our DNA strands, and also, of course, my supervisor Stefan, for giving me the opportunity to work on this project, and all the other students that have given us a lot of fun over the years. We can see here Annika, Maria, and Niklas. But there have been others that I haven't mentioned here. Yes. Thanks. So I hope we have a little bit of time for questions. Yes?
So you say one of your limits, especially getting them close enough together to interact, and that could influence basically as well the yield of how much reactions you have. Why don't you just use longer strands? Like make your anchors are now 17 base pairs, and like if you just have longer ones, wouldn't that allow longer distances?
Let me just quickly go to the slide. So if we want to simply pre-assemble liposomes containers, there could be other applications of that. We could use anchored DNAs that hybridize in this fashion. And there with length we can control the distance actually. If it hybridizes in a zipper-like fashion, we always want to get them as close as possible. So here the final distance depends mostly actually on the of course, the strand needs to hybridize strongly, but the minimal distance depends on kind of the exact chemistry of the linker and the head group of our anchors in the membrane. So, and actually we've played around with that quite a lot, and as I maybe mentioned, the presence of this linker, presence or absence of this linker gives actually a huge increase in an infusion, where actually having a small linker increases the efficiency of the content mixing a lot. Is that
Yeah, so do you have any DNA binding events that don't result in fusion?
I think we have many DNA binding events. So, for the first of all, for example, for the we have the cascade here. For that one, we use about 40 copies per liposome. So, for 2 to fuse, I think at least 10 or 15 need to hybridize to kind of make when you kind of make this cross section stable enough, kind of need to have zippers kind of, I guess, around, all the way around kind of this cross section between 2 liposomes. And for that you need at least a few DNA strands, maybe 8 or 10, depending also on the size of the liposome. If you want to use a GOV to make stable adhesion, you maybe need 1,000 molecules. For if we could make our liposomes much smaller, maybe 40 nanometers, like actually they are in neurotransmitter release, that range, then maybe 5 to 10 DNAs would be enough. Yeah. But controlling the production of nanoparticles is actually an entirely different issue. There have been recent reports on using kind of a DNA ring, kind of re-template the size of these things, but it's, yeah, again adds another level of control. So, yes?
So a lot of in these chemical reactions you want to make using this fusion, often I guess there's temperature changes and require specific temperatures. How sensitive is your system to temperature changes?
I think this is a very good question. We tried to do fusion at room temperature for around 2.5 years. And because that's the temperature where the DNA hybridizes best, at least compared to higher temperatures. But we actually do all of these experiments at 50 degrees, which could be a problem for some enzymatic reactions. On the other hand, after 5 minutes at 50 degrees, more than 90% is basically fused. So most enzymes would tolerate that. In case of chemical reactions in water, most of them would not be so crazy exothermic as you need to kind of freeze the system to prevent then you probably, yeah. But you could do it, of course, then you need to control something more, kind of where you need your hands. You could do a fusion step, for example, at 50 degrees, and then a reaction step at 37 or 20, or maybe even 60 or 70, depending on how likely your contents are to penetrate the membrane and go to the outside. That scales with temperature. So the higher temperature we do the reaction, the more leaky the liposomes become. But there actually nucleic acids come to our advantage again, is because they are actually polyanionic molecules, and all the phosphate groups in the backbone kind of add a lot of negative charges to the surface, of course depending on which salts, but with monovalent salts there's a negative charge on the liposomes. And that negative charge actually increases the more liposomes you fuse, because more DNA comes together. And especially also because the double-stranded DNA, once it's on the same membrane, would likely kind of really extend from the membrane, giving kind of a negatively charged protection layer, which actually prevents leakage to to some degree, as we saw in some of our controls. Yeah, it's too far away now. Yes?
Maybe important to mention that it works fine at 37 degrees. So enzymes
Yeah.
are perfectly compatible. Only the extent of fusion is reduced, but at best a factor of 2 or something. So there is still massive content mixing at temperatures which are perfectly fine.
You can actually see how the leakage results in signal increase in absence of DNA or anchored DNA, and during the fusion this is actually less. There's a slight increase in the beginning, and if we have completely non-complementary DNA on the surface, then we prevent most leakage even at 50 degrees. These types. Alright. I think maybe a
I
last
just have a
question.
small question.
Or, yeah.
About the FRET study,
Yeah.
have you done it? Have
Yes.
you done any FRET?
Yeah.
But then have you put the donor and acceptor in the same vesicle? Because it shows here that you put it in
Yes.
the same, but wouldn't
Yeah, I
you put
will.
it in
I
different
can
ones?
quickly go there. Let me see. Yeah. So the FRET, actually what you're monitoring is recovery of the FRET donor. So we monitor the fluorescence recovery of the FRET donor. So we have one population where we have two lipids that are a FRET couple. They initially they are both on the outside and the inside, and we have destroyed the outside dyes with reduction, rinse it, and then we have kind of asymmetrically FRET labeled liposome, which if you look at a FRET channel would have low fluorescence, would have higher fluorescence here than here, but if you look at the donor fluorescence, you have a low donor fluorescence because it transfers its excitation energy to the acceptor, but after kind of dilution on the surface, when the inner surface becomes bigger, then the signal for the donor recovers. Yeah.
But why wouldn't you put them the donor in one and the acceptor in the other type of vesicle, and when they fuse, you will see a FRET effect? Anyway, we can
That's a good
discuss this
idea.
afterwards.
Yeah. We also talked about doing that with kind of encapsulated DNAs that then produce a FRET effect. Yeah, that would be a very good proof of that the vesicle hasn't just like grown spontaneously somehow or swollen or yeah. But I mean, sometimes then there are historical literature reasons why you use a certain assay. Yeah.
And also, if you had them mixed, each donor in one population and acceptor in the other population, it would be difficult to discriminate between donor-donor fusion and acceptor-acceptor fusion. And that problem is circumvented here, I guess.
That might be true, yes.
Yeah, I was just wondering, don't you think if you are not using oligonucleotides about the fusion, if you have two functional molecules that will react, let's say quick chemistry, the fusion will be more efficient and less costly?
Yes, but then the problem is that we can only encode one step or as many orthogonal chemistries as we can devise. So we can do one fusion based on some click chemistry, and then the next fusion based on a amide bond, and then maybe some Wittig reaction.
That's a disadvantage, yes.
And then so we try to come up with something that is flexible so we can. Yeah, I mean, after a few fusion steps, we probably would need to add some DNA to kind of start from scratch, because else they will become so negatively charged that they could not fuse anymore. Yeah. Well then, thanks again.